Hi, my name is Meaghan Gade, and I am a PhD candidate at Ohio State University, and this is my 4th year here at Highlands Biological Station. I'm studying salamanders, plethodontid salamanders. These are lungless salamanders that are found throughout this region, and here in Highlands is a really good base camp for us to be studying this sort of salamander. They're really interesting because they actually don't have any lungs, so they have to be living in really cool and moist environments, which is why high elevation areas like Highlands are great for these sorts of salamanders. So I've been working here for 4 years, been funded through a grant and aid by the foundation. And for the past 4 years, I've been collecting long-term data on population level demographic information on these salamanders. So understanding what their survival is like, how quickly they grow, if they move, and all these sorts of different parameters. And so the way we do that is we go out into the field, and I have an area that I go to specifically every single year, and I capture every single salamander that I can find. We bring them back here to the lab at Highlands, and actually I can anesthetize them using a little bit of Oragel in a solution of water. They'll fall asleep, and then I'm able to tag them using a little fluorescent plastic that's a specific color. And so with that color, every time I recapture that individual, I know who it is. So it's an individual maybe has 2 red marks in it. So every time I catch him or her, I know that that's that individual. So I can track if it's moved, how fast it's grown, and all these different sorts of things. I'm focused on the Redlegged Salamander, Plethedon shermani. And we actually don't have any of these in the Highlands area. So I go out to Wayah, which is about a 45-minute drive past Franklin. And these salamanders are really interesting. I'm gonna try to get him out of his bag here. Because they have, like I said, they're lungless. And let's see. And they have these really bright red legs. This is an adult. They'll get a little bit bigger than this, but this is about a good adult size animal. And you can see those nice bright red legs. Here in Highlands, you'll see a really similar looking salamander to this, and they don't have the red legs, and instead they just have these really gray cheeks, and that's called the gray cheek salamander, Plethedon metcalfi, and they're closely related to Plethedon shermani here. And so, like I said, we go out to the Mark-recapture plots and we catch every individual that we can in that given area. So when we bring them back to the lab, we want to put them to sleep so that we're able to manipulate and measure them with a little bit more care. So this right here, this is just a solution of Oragel and water, and we can just drop them right in there, and it'll take about 3 to 5 minutes for these individuals to fall asleep. So after about 3 to 5 minutes, the salamander is asleep, so they're actually pretty easy to manipulate at this point. And the reason we do this is just so we can get really precise measurements, and it makes it easier to tag these salamanders. And so what we do is we flip them on their back, and then we're using a plastic elastomer, and we mark them with different colored marks. And so this individual has a yellow mark on the top arm there, a blue mark, another blue mark, and then an orange mark. So every time we catch this animal, we can read that mark and we know who this animal is. And so I've actually captured this animal 3 different times over 4 years. And so I'm able to track growth, I can see where it's moved, all different sorts of things. Over the course of 4 years, I've captured about 2,300 salamanders, and I've recaptured at about 35%. For this species here, the Plethedon shermani. These animals will wake up after another 5 minutes, so we usually dunk them in just some fresh water to wash off the Oragel, and they'll wake back up, and then we'll head back out into the field tonight and put him back right where we found him. So these animals are fully terrestrial. They hatch out of eggs as just little miniature versions of the adult that we just saw. And they'll actually live their entire lifetime in an area maybe about 3 meters squared. And so at my Mark-recapture plots, we have the areas sort of marked out in 1 meter square blocks. And so this individual that we've looked at today, over the course of 4 years, has only moved 2 different blocks. So we've only found him in 2 blocks, so he's only moving about 2 meters squared. And that's pretty common with these terrestrial salamanders, that they don't tend to move a whole lot.